All right, so let's talk a bit about mutations. Mutations are any changes in the nucleotide sequence of DNA or RNA. They can occur spontaneously, or they can be caused by mutagens. Mutagens are physical or chemical factors which can cause a change in the nucleotide sequence. Some mutagens, examples of mutagens include things like UV radiation, gamma rays, X-rays, cigarette smoke, some viruses, insecticides, pesticides, etc. So let's talk about some kinds of mutations. Let's first of all talk about point mutations. You'll sometimes hear these called substitutions. They're a type of mutation in which a single nucleotide is changed to another incorrect nucleotide. So this changes just one codon. We'll talk about 3 different types of point or substitution mutations. One type is a silent point mutation. In these mutations, even though one of the nucleotides is changed, the resulting nucleotide still codes for the same amino acid. So therefore, the amino acid sequence doesn't change, the protein doesn't change, and even though a mutation happened, there are no negative effects. Next, we'll mention a missense point mutation. These happen when a nucleotide is changed, a single codon is changed, and that causes a single amino acid to also be replaced by a wrong amino acid. So this type of mutation can be dramatic, can cause dramatic effects, especially if the chemical properties of the original amino acid and the new amino acid are very different. For example, sickle cell anemia, the blood disorder, is caused by a point mutation in the gene for hemoglobin. During this mutation, an adenine nucleotide is replaced by a thymine nucleotide. This causes the amino acid glutamic acid to be replaced with the amino acid valine. And those 2 amino acids have very different properties. So this causes the resulting hemoglobin to have a very different shape than normal hemoglobin. And it can lead to things, symptoms in sickle cell anemia like problems breathing, poor circulation, all kinds of pain, dizziness, fast heart rate, delayed growth, early death even. Here's an example of that. So here's the actual normal DNA sequence for hemoglobin. And here's the normal amino acid sequence for normal hemoglobin. Here we see the DNA sequence for a person with sickle cell anemia. And notice that the only change is in that nucleotide. That causes the substitution of one incorrect amino acid. But because the properties of these 2 amino acids are very different, the resulting hemoglobin and ultimately the resulting red blood cells can have very different structures. While we're here, let's also talk about nonsense point mutations. So this is when a single nucleotide substitution causes a codon which normally codes for an amino acid to become a stop codon. So this causes the termination of translation to happen prematurely, and it's going to cause essentially none of the functional protein to be synthesized. These are pretty much always going to be very serious disease-causing mutations. So those are point mutations. They involve a substitution of a single nucleotide. Well, let's also talk about frameshift mutations. You may hear these called deletions or insertions. These are when one or maybe several nucleotides are either deleted or inserted. These mutations can introduce premature stop codons, and they can change the entire sequence of the amino acid chain in points after the mutation occurred. These mutations are usually more severe than point mutations because they cause extreme changes and they affect multiple amino acids. So here's an example. So here's the normal DNA. Here's the normal amino acid sequence. So now we get an insertion. And remember that codons are segments of 3 nucleotides. So because we inserted this new set, everything got scooted over and the reading frame got changed. So now instead of getting MET, ARG3, and PHEN, we get a different combination of amino acids. Here we get the same kind of idea, except now we get, instead of an insertion, we get a deletion of a pair. Well, that scoots everything over, and now, again, we get a completely different set of amino acids at points past the mutation. The mutation happened after the methionine. So methionine is still correct, but everything else is different because of that change in the reading frame caused by this frameshift mutation. So another example of, an example of a frameshift mutation is Tay-Sachs disease. This is caused by a frameshift mutation in the hexagenes, which are genes that code for an enzyme called hexaminidase A, which helps to break down certain kinds of lipids. Because of this frameshift mutation, the insertion of a TATC, the shape of the enzyme changes, and therefore that enzyme isn't functional. And these nucleotides, or these lipids rather, that they would normally be broken down, accumulate in the brain. And because of this accumulation of lipids, people ultimately experience deafness, progressive blindness, and lots of other symptoms, but ultimately they experience death typically before the age of 5. It's a very miserable, serious condition. So over here, let's also talk about gametic versus somatic cell mutations. So mutations which occur in the germ cells, the cells in your body that can undergo meiosis, or the gametes, those can be passed on to the offspring. They can lead to evolution. On the other hand, mutations that happen in your normal body cells, the somatic cells, those mutations are not passed on to the offspring, and they impact only the person with the mutation. They don't contribute to evolution. They're not passed on to other members of the population. All right, that's going to be it for mutations.